congratulate Professor Sri Kumar Apte for receiving the Professor Vishwa Nath Memorial Lecture. Due to COVID-19, this got delayed. So he will be speaking on, the title is here. Oh. World's most radiation-resistant bacterium, Deinococcus radiodurans, and what can you do with it? So before he goes into his lecture, I would like to tell something about the lecture. This Professor Vishwa Nath Memorial Lecture was established in 1992 to commemorate the memory of Dr. Vishwa Nath, an eminent zoologist and a distinguished fellow of the Academy, out of an endowment made by his students, family members, and friends. The lectureship is awarded once in 3 years since 1995 for outstanding contributions in the field of biochemistry and biophysics. So the past awardees are Dr. Lalji Singh, K. Muniappa, Siddharth Roy, P.K. Das, and Dr. Shubhratu Sinha. So, so with that, I would invite Dr. Apte to come and deliver his award lecture.
Well, good afternoon, everyone. And I would like to begin by thanking INSA, first of all, for considering work of my colleagues worthy of this award, and more importantly, for making this pre-pandemic award actual reality after the pandemic in this anniversary general meeting. Most biologists here know me as a worker on cyanobacteria, the most ancient living organisms on this planet. But last 10-12 years, we have been also working on this organism, Deinococcus radiodurans, which Guinness Book records as world's most radiation-resistant bacterium. And for today's presentation, I have chosen our last decade or so's work on this organism. So this is how the Deinococcus looks on a Petri plate. Deinococcus means strange red berries because that's how the microbiologist thought of it on the plate. It is strange in more than one ways, including its habit of growing 4 cells together as tetrads. But the most impressive thing about it is its extreme radiation resistance, which, as you can see here, compared to the survival of humans and 3 other organisms with increasing doses of radiation, the humans being almost on the y-axis, followed by world's most hated animal, cockroach, and E. coli, the model organism for microbiologists. Deinococcus survives up to 6 kilograde dose of radiation as if it has never been exposed. Its survival at that dose is 100%. For those of you who are not familiar with what 6 kilograde dose means, 6 kilograde is the expected dose within 100-meter radius of a Hiroshima-sized bomb. So nobody looked for this organism then, but it may have survived just that. So the question as to what can you do with such an organism has been there right from the beginning, and 2 ideas were always there. One, because of its extreme radio resistance and the ability therefore to survive DNA damage, oxidative stress, and so on and so forth. One idea was to look at some novel mechanisms that it may have for surviving such high doses of radiation, for highly proficient DNA repair, and oxidative stress tolerance. But the other idea was, of course, to try and develop some biotechnologies for high radiation environments, particularly in the nuclear field, where I come from, Bhabha Atomic Research Center, for example. And those ideas still continue, although now several non-nuclear applications are also thought of. So in the limited time that I have, I will try and show you glimpses of some of these. So when I say that at 6 kilograde the organism has 100% survival, it's not that it doesn't suffer any damage. There were papers in Science and Nature 20 years ago saying, does this organism wear a lead shield? Lead being an effective blocker of radiation, but that's not the case. At those kind of doses, its DNA literally goes to pieces, completely shattered. This is an optical image of the DNA damage. but you just take out the cells, wash them, put them in the fresh medium, and let them grow over a period of time. It will put each of these pieces in the correct place, in the correct orientation to stitch back its original chromosome, and then its growth starts once again as if nothing has happened. So that's the remarkable capability that this organism has, which no other organism has. Impressed by this, and also by the fact that pond waters in some places in US suddenly got radioactivity because of the nuclear stockpiles that were stored there during the Cold War period, which leaked, and radioactivity leaked out. And when they looked for what is surviving in such lakes, if at all, they found Deinococcus there. Originally, it was discovered from irradiated meat cans that were used as food for American soldiers fighting in Europe. So it has a long history of surviving radiations. So it was the US Department of Energy, of all the other funding agencies, that funded genome sequencing of this organism to know what is so special about it. And a few things that are special about it are shown here. The genome size is small, just about 3.3 megabases, smaller than E. coli, but it has 4 different pieces of DNA that you are seeing here, 1 large chromosome, 1 smaller chromosome, 1 megaplasmid, and 1 small plasmid. But interestingly, each of these pieces of DNA are present in a cell in 4 to 10 copies, so at a particular time it may have 40 pieces of DNA in the cell. And that offers it an advantage that there is a multiplicity of not only genomes, but a redundancy of genes, and in response to radiation damage, some or the other gene copy gets damaged, but some copy survives, and it simply copies and pastes that by homologous recombination and manages to survive. Only beyond a certain dose, when no good copy is available, that it starts showing radiation sensitivity. Now, other details are here. More importantly, of the 3,000 odd genes that were identified at that time, 2,000 are known genes, which were also there in other organisms, but close to 1,000 genes, nearly 30% or 1/3 of its genome, were unique genes not found in any other organisms. And the question was that its remarkable ability, is it because of already known genes or because of those unknown genes which probably hold a secret, and that's what some of the earlier attempts were made. This is a very congested slide, but all I want you to see are the red colored names. This just gives you the total picture of DNA repair pathways in an organism like this. One would expect that it would have everything that other bacteria have, plus many more, but strangely, the red ones that you see here are genes which are normal DNA repair genes in other bacteria, but they are absent in this organism, including the orange ones at the bottom, which are responsible for surviving ultraviolet radiation, for example. So that was a big surprise as to why this organism lacks some of the routine DNA repair genes in other organisms. Of course, there is a multiplicity of other kind of genes, such as polymerases. It has even human-like polymerases. It has several DNA glycosylases. It has several DNA cleanup genes, and so on and so forth. But absence of certain genes was a big surprise. So what about the genes that were present, and what about the genes that were absent? Some earlier work was done. Over here, for example, shows you the survival of Deinococcus versus, again, very high doses of ionizing radiation. And the curve at the top is the wild-type Deinococcus, which, as I have shown you earlier, survives very well. But over there are 4 different mutations in individual genes. RECA, POLE, UBRA are prominent DNA repair genes in any bacterium. And if you just mutate this one gene out of 3,000, Deinococcus becomes almost as sensitive as E. coli, indicating that just this individual gene holds the secret of this organism's radioresistance. But that's not the case, because if you take this gene from Deinococcus, put it in E. coli or any other organism, it does not increase its radiation resistance even 1 bit. So the conclusion which you see at the bottom was that these genes are important, but their contribution is context specific. In the Deinococcus context, they work extremely well, but you move them elsewhere, they are not so effective. Well, what about the genes that were absent? Why should Deinococcus lack some of the important DNA repair genes? Again, one example I am showing you. SBCB, which codes for exonuclease, a very important gene for DNA repair, is absent in Deinococcus. So we asked the question, what will happen if we take this gene from E. coli and put it in Deinococcus? And very strange thing happened. The otherwise radiation-resistant Deinococcus suddenly became radiosensitive. So a gain of function, a gain of a gene made it radiosensitive, and all the other details that you see there on the right are the DNA and damaged DNA following irradiation run on a gel, electrophoresed on a gel to see its size. To start with, the unirradiated DNA becomes shattered. You see very small pieces of DNA, but allow it time and it will stitch back it. Within 24 hours or so, it will get back to normal unirradiated level of DNA. But when you transfer this SBCB gene from E. coli into Deinococcus, you can see in the second half of that gel, it loses its ability to stitch back this DNA. So in not having some important DNA repair genes has been a gain for Deinococcus and its DNA repair efficiency and therefore DNA repair ability. That's also the case for some other missing genes, but I think one example is enough for now. What about proteins, which are the other important biomolecules which actually regulate whole life function at all times? So a strange result was that if you irradiated it at 6 kilogray, separated the proteins on two-dimensional gels, what you are seeing here are those individual protein molecules indicated here as spots. You can see that the two halves of the gel are almost identical. First one is unirradiated, second one is irradiated at 6 kilogray, as if nothing has happened to it. Now this excited a lot of crystallographers, and they thought Deinococcus has some very highly radiation resistant proteins, but which was not the case. If you take the second set of cells, wash them, again start putting them in the fresh medium to grow and see what happens, interesting things happen. You can just see a time course of the recovery process, 2 hours, 6 hours, 12 hours, compared to the 0-minute profile. At 2 hours, you can see that a lot of proteins are degraded. And then they start coming back after 6 and 12 hours. And this led to the discovery of a phenomenon which we call protein recycling. Protein recycling happens in cells all the time. Each protein has a lifetime beyond which it's turned over. But here, in response to irradiation, the proteins which get damaged in the form of carbonylation or nitrosylation are selected and degraded first, and then they are resynthesized within a short time, and the recovery has to wait till both the DNA is repaired as well as these proteins are repaired. So it had this novel phenomenon of protein recycling during post-irradiation recovery. In fact, for such recovery, you will need a lot of DNA repair genes, and when we looked at the protein profiles of these organisms over a short period of 4 to 6 hours, we found that several DNA repair genes which were at low level at 0 hour increased over next 3-4 hours and then came down. And after these 3-4 hours, of course, normal growth started, indicating that not only these genes were expressed at a higher level so that they contributed the DNA repair activities, and then the Deinococcus recovered fully. So that was true of both the DNA repair genes as well as oxidative stress tolerance genes shown here. Now, why does it happen, or how does it happen that an organism like this can express genes, required genes at a higher level after such a massive dose of irradiation? Now, that took about 10 years to discover, but one of the early discoveries that were made way back in 2003 was this gene from Hua's group in China, the PPRI gene. PPRI stands for pleiotropic protein response inducer, which means post-irradiation, it induces the expression of several proteins. And Hua had randomly mutated Deinococcus and identified this gene to show that when this gene is mutated, no genes are expressed post-irradiation. So it acted like a molecular switch. And as you can see here, as a consequence, not only Deinococcus becomes radiosensitive, it fails to induce expression of all those important DNA repair genes. So somehow this gene was very crucial for its post-irradiation induction of DNA repair genes as well as for radiation resistance. But how does it happen took another 10 years to discover. Somewhere around 2009, 2 more Deinococcus species genome sequence was available, and the groups listed there, particularly Makarova et al., wanted to know what is the minimal number of genes that is required for such radioresistance, what they considered as minimal genome for radioresistance, by comparing the genome sequences of these 3 or 2 Deinococcus strains. And what they found was very interesting. They found close to 125, 130 genes, each of which they mutated and found that the organism became radioresistant. But upstream of each of those genes, they found this particular motif. 18 base pair palindromic sequence. It was a poor palindrome in the sense only the 5th G and 13C were absolutely conserved, and the first 2 and last 2 bases were also conserved, and they were palindromic in nature. Rest of the sequence did not matter much, but these had to be absolutely conserved. Now, since this was present upstream of all DNA repair as well as desiccation resistance genes, they named it radiation desiccation response motive, in short RDRM. This was only a deduction out of the comparison of genome sequences. Promptly next year, our laboratory investigated if this was indeed so, whether this indeed contributed to the DNA or desiccation resistance. And as you can see here, that we did using the single-stranded DNA binding protein genes promoter for this. The SSB gene that you see there in yellow has a upstream sequence comprising of 2 RDRMs, RDRM1 and 2, and we progressively deleted each one of them to see what happens. We also tagged it to a reporter gene like acid phosphatase 4N, which on plates makes green colonies when it expresses acid phosphatase. And what you see here, that before and after radiation, those colonies became less green or more green, but more important result is on the right side gel. We found that in response to exposure to gamma rays as well as mitomycin C, both of which cause extensive DNA double strand damage, the SSB gene promoter was highly induced. And when we progressively deleted each RDRM, the expression decreased less and less, maximum in the last lane where you have both RDRMs, then in the middle lanes where you have only one RDRM, and the radiation-induced expression was lost when the RDRMs were deleted. Now, based on this, we embarked on a very ambitious project to characterize all Deinococcus promoters. Close to 21 different promoters we took. And what we did was to tag it to green fluorescent protein gene, because then we could visually, microscopically examine each cell of the tetrad to see what happens. And this is what happens. So in the absence of radiation, you will see their expression at low level in the left panel. And on the right, the same cells expressing these promoters would show much higher level of green fluorescence. And over here is a quantitation of some of them to indicate that in some cases up to 25, 30-fold enhancement in their expression took place following irradiation. Now the mechanism underlying this, if it was RDRM, which was already proven, was discovered later to find that this protein called DDRO is a repressor of all these genes under normal conditions. Normally it binds to RDRM and blocks the expression of all these genes or keeps them at a very low level. Post irradiation, this DDRO is cleaved by another gene called PPRI, which I described earlier as a molecular switch. So it took 10 years to discover its role. It cleaves DDRO, and then all these 125 genes get induced, and that promptly happens following radiation damage and not before that. Before that, they express only at a low level. So organism has a very efficient way of controlling the level of these genes, normally at a level where it is required, but post radiation damage, particularly DNA damage, it has also a way of enhancing their expression several fold, getting many higher fold levels of DNA repair genes and undertaking DNA repair. Now we proved it in vivo also by taking E. coli as an example. E. coli neither has RDRMs nor has PPRI. So we cloned Deinococcus DDRO in E. coli. The top slide shows that. So DDRO protein is expressing. And then we moved all these 21 Deinococcus promoters tagged to GFP into E. coli and showed that the blue bars show their level in the absence of DDRO, and the purple or violet bars show their level after DDRO, showing that the DDRO indeed is a repressor of these genes even in E. coli. You can move it to another organism and you still get that effect, saying that the DDRO is the in vivo repressor of this. And don't have PPRI anymore, and you show that in such a Deinococcus cell, the radiation has no effect. Before or after radiation, level of each gene remains same. So the model is proven both in vitro as well as in vivo, and that's a very robust DNA repair gene induction response in response to radiation. Radiation inductive gene expression has been reported in many organs, including humans, but such a robust response where you can see this promoter activity very nicely, monitor it, and we have even used it in certain biotechnologies. I will show you an example. This is probably the only example that I know of. So I'll now return to the question as to what can you do with such a wonderful organism. Find that it's extremely high doses of radiation, or for that matter, any genotoxic stress, ultraviolet radiation, desiccation, nutrient starvation, all of them it resists very well, because anything that damages DNA, it has a way of inducing DNA repair mechanisms and tolerate that. But what can you do with such an organism? Now, one of the things that everyone thought of in the beginning, in fact, the very first paper after the genome sequence was published was from US, which was to show that the organism can be used for degrading toluene and mercury, which are present in the nuclear waste, because that was one of the desirable things to do. Radiation was very high in those environments. This organism survived such levels of radiation, so you may as well exploit it for that purpose. We thought of doing something else. Radioactive waste, after processing, has a lot of uranium in it. Well, it traces of it only, less than a millimolar, but it keeps decaying and produces some very, very long-lived isotopes or radionuclides. So if you can get rid of these traces of uranium from the radioactive waste, it becomes much easier to dispose. And we thought we will make use of this bug for that purpose. Now, uranium is more stable as phosphates. Uranium phosphates are the most stable form of uranium, and so we thought we will precipitate this uranium out of such low-level radioactive waste by employing phosphatases. So for which what we did was we chose an acid phosphatase that lives in the periplasmic space of the bacterium. In this case, the bacterium is not Deinococcus. The gene has come from Serratia. And then we have cloned it in Deinococcus. Now in the periplasm, if you give it an organic phosphate like beta-glycerophosphate here, it will cleave it to produce phosphate or phosphoric acid, and that phosphate, if you provide uranium outside, it will precipitate that phosphate, and that phosphate gets deposited on the membrane. Now all of this has importance in bioremediation, as I will show you later. Another important thing is because of the acidity that's produced there, we put an indicator dye, pH indicator dye, which is colorless at neutral pH, becomes green at acidic pH. So it becomes very easy to monitor such things. I'll show you that in the next slide. So that's the approach. Clone a phosphatase gene, give it an organic phosphate, and the phosphate produced will be used to precipitate uranium. And that's a summary of some of the early work where we engineered Deinococcus as well as E. coli to precipitate uranium using such phosphate. Now, precipitating uranium as phosphate is not so significant, but being able to do it at less than millimolar concentration is significant, because no physicochemical methods can precipitate uranium at that concentration. So over here you see with increasing time, more and more phosphate gets precipitated. By about 8 hours, more than 90% uranium phosphate gets precipitated and removed from the waste. Both E. coli and Deinococcus can do it, but once you irradiate at 6 kilogray, only Deinococcus can do it. E. coli goes to pieces. It in fact cells completely disintegrate, it loses the ability, and under high radiation environment, E. coli cannot function. So that sort of proves the point that why it should be engineered in Deinococcus. And over here is the assay plates. The orange color is the normal color of Deinococcus, and when you engineer it to produce phosphatase, and then you add that indicator dye, those colonies which are positive turn green. So that's a very easy assay, and eventually you see it like this. Now this is all done using spectrophotometric methods, and somebody commented that we don't see any uranium. Normally those of you who may have worked with uranium when it is made, it's big yellow colored cakes are available. So why can't we see that uranium in these kind of assays was the question, or rather seeing is believing. So we did that on columns. If you load, this is the normal column with normal Deinococcus color, and that's post precipitation, the entire column turns yellow because of uranyl phosphate. We then also did scanning electron microscopy and transmission electron microscopy. So the tetrad, Deinococcus tetrads, after they have precipitated uranium, gather such fluffy mass all around them, and if more and more time, the mass keeps growing because the precipitate gets attached to the membrane, and just that serves as the site of nucleation, and precipitate just grows. On the right hand side is the EDX to show you the uranium peaks compared to the unexposed. The exposed one has several uranium peaks there, indicating the identity of this particular precipitate. And in transmission electron microscope, you also see all that uranium sticking around the cells. Now, uranium being a heavy metal, after 2 or 3 hours, it makes the cells so heavy that they settle down, and that's a big advantage for bioremediation, because in a 30, 40, 50 liter waste, in 3 hours, if entire uranium gets adsorbed onto the cell surface and settles down, you simply decant the top supernatant without having to centrifuge, etc. and you collect the cell mass, you recover all the uranium, which you can do all the way. Mind you, the purpose of this is not to extract uranium to be used as fuel. This is nanomolar, micromolar, millimolar quantities we are talking about. The purpose is to remove it from radioactive waste so that it can be safely disposed. Now this was all done in acidic pH where the uranium is normally extracted. Somewhere around 2008 or 9, alkaline methodology started because India also discovered uranium mines in Tumlapalli in Andhra Pradesh, and where the alkaline process was to be used. So the idea was whether we can do it in alkaline pH. Now alkaline pH has a serious, before I go there, the radiation induced promoters that I talked to you about, we tried to see if we can exploit them. And at two different levels of radioactivity, in low level waste and high level waste, we use such promoters to show that with radiation they do get induced. With irradiation their phosphatase activity increases several fold compared to unirradiated controls, and you can indeed get much higher levels of uranium precipitated right in radioactive waste where no other organism will function, no other promoters function, but these being radiation induced promoters work there very well. So coming back to alkaline pH, bacterial alkaline phosphatases have very, very low specific activity. That's why in molecular biology, we mostly use eukaryotic alkaline phosphatases. So by 1.5 years effort, we found a bacterium, which is Novosphingobia, which produced an alkaline phosphatase extracellularly of a very high specific activity, more than 1500 units per minute per milligram protein. And that's the initial work to show you that it has two acid alkaline phosphatases at pH 9. The gene was cloned by transposon mutagenesis. I will not go into those details, and cloned into first into E. coli. So that shows you the original strain. It produces green colonies because of acid phosphatase, and the alkaline phosphatase comes out and gives this blue halo around the colonies. That's a mutant of it, transposon mutagenesis, where the acid phosphatase in the retained, alkaline phosphatase is lost, and then you can clone it in E. coli or Deinococcus to get required expression. So we could get 47-fold higher expression of this enzyme after we cloned it in E. coli and Deinococcus compared to normal strain, which takes 7-8 hours to precipitate 90% uranium. The engineered strain could precipitate it just within 1 hour, and that's because Deinococcus seems to be a very good host for this particular enzyme. It has a more optimized codon usage than in fact Novosphingobia. So it worked very well. When we sent this paper out, chemist was a reviewer, a uranium expert was a reviewer, and no offense meant to chemist, but he said that I don't understand your enzymes, but I know one thing: if your alkaline condition, your solution is in uranyl carbonate, it's impossible to precipitate uranium as phosphate unless you generate 1000-fold excess of phosphate over carbonate. So please do an XRD and show me it is uranyl phosphate, which was a good suggestion because when we did the XRD, the precipitate was indeed found to be uranyl phosphate, that particular species with that composition, and it also identified it from literature as meta-autunite or Chernikovite, which is named after the person who discovered it. We also learned that this precipitate will fluoresce intense green if we shine UV on it, and that's what you see here. So that proved its identity. This fluorescence reaction also gave us additional assay for doing online monitoring of uranium precipitation in radioactive waste, near which you cannot go, but a spectrophotometer can be, a fluorescence spectrophotometer can be put there, and you can monitor all this. Now the one difference here is that unlike the acid phosphatase which I showed you earlier, this precipitate was not cell bound, but it was in the solution. You can see those uranium crystals there. So it required a different approach for recovering this. These cells were immobilized first in beads like these. So there's empty beads, Deinococcus containing beads, and then all the precipitate there which could be detected by fluorescence. So over here you can see beads control. They also absorb some amount of uranium, but post precipitation all the beads contain more than 80-90% of uranium, and so you have another assay for detecting uranium precipitation. The last but not least, the question was, after imparting this extra work on this Deinococcus, does it compromise its radio resistance? So which was also tested. No difference in its radio resistance before or after giving it the ability to precipitate uranium, and such cells even after 6 kg exposure continued to precipitate uranium very well. So this is a self-sustaining technology which has been used on a small scale in our waste management division. Let me now turn to some non-nuclear applications, as I said. the kind of people who got interested in Deinococcus because of its strange and interesting abilities range from space biologists to cancer specialists to nanoscience people, and I'll just show you some examples of that time. So over here is a work reported in 2017 and subsequently several other papers where space biologists got interested into it because they thought the organism was so extremely resistant to all kinds of stresses that they could use it. So they prepared a biofilm of Deinococcus on the top or planktonic free living cells loaded both on the membranes of some kind to provide a substratum and then expose them to all kinds of conditions, prolonged desiccation, extreme temperatures, vacuum, and simulated Martian atmosphere. I will not describe that to you. You can look at this paper for details. And last but not least, ultraviolet radiations, and created combinations of these to either simulate Martian conditions or desiccation or space conditions, and tested them to find that the biofilms survived much better. The entire green color is showing you the viability, and the red color at the bottom, after they are exposed to UV, they lose their viability, but they continue to show this orange color because of the orange pigment, which is a carotenoid in Deinococcus called dinoxanthine. And dinoxanthine subsequently has become a very important molecule. It is used as a marker for space biologists in their space missions to see how this molecule survives extreme conditions of space. So we will hear more about that in near future. But then this molecule is also found to be a pro-oxidant at a certain concentration and an antioxidant at other concentrations. So it has been tried by cancer people for cancer therapy. So over here are 3 different cell lines where the cell viability by increasing concentrations of, these are cancer cell lines. Viability is found to be decreased, number of apoptotic cells found to be increased, some cancer markers like BAX expression was found to be increased. So in general, the different concentrations and different regimens of dinoxanthine application have been found to be good for repressing or regressing certain kind of cancer in cell cultures or even in some in vivo applications. There are many, many more papers of this kind. I have just chosen one to make the point. Nanoscience people also got interested in it for 2 reasons. One is Deinococcus has a surface layer called HPI, which is a self-assembling nanomaterial. That's how it organizes itself. So you purify it in solution, just leave it there for an hour or 2, and it will make a nice membrane by self-assembling itself. And very discrete, well-spaced units it has, which has some interesting properties for nanobiology. Now, first of all, we use this to tag our phosphatases to surface layer and express it on Deinococcus surface, as you can see. And this also enhanced the uranium precipitation ability several fold, because now the active molecules were being expressed on surface. But nanoscience people who wanted to grow nanowires and increasingly use biological materials for green ways of producing nanomaterials found a very interesting applications. They just took this S layer, which self-assembles as a nanomaterial and has well-defined, well-spaced units. They did all this, which I am not describing. This is a way of growing nanowires, vertical nanowires on such surfaces, and found that they not only grew very rapidly, grew in desired way. In general, they start thick and then taper off. Here, everything was of uniform length, uniform height, uniform spacing, and the credit for that they gave to this well-spaced nanostructure that was available from the surface layer of Deinococcus. So a lot of work has gone in this direction also. People have also tried to make nanoparticles, metallic nanoparticles, bimetallic nanoparticles, and multimetallic nanoparticles using Deinococcus extracts in this particular fashion. Over here is a picture of silver gold and gold silver nanoparticles, which were prepared again using Deinococcus, and showing you the dual abilities of one, the antimicrobial properties. Silver nanoparticles, as you know, work very well as antimicrobials, as well as ability to reduce certain dyes. In this case, 4-nitrophenol was reduced using the gold part of the bimetallic. So a lot of work on constructing bi and multimetallic noble metal nanoparticles and putting them to these kind of applications. This is a 2017 paper. In 2021 and 22, another 70, 80 papers on these lines have come out. Last aspect that I am most impressed with was an inherent ability of Deinococcus that it protected its proteins much better than it protected its DNA, for reasons that were found to be in the extracellular medium. When you grow Deinococcus in a culture medium, after 4 or 5 days, when it has used up all the nutrients, if you just take that culture filtrate or the spent medium, It was found to be very rich in antioxidants because Deinococcus secretes certain peptides. Deinococcus is grown with excess manganese, and the medium has lot of phosphate, and all this resulted in very strong protein protecting antioxidant solution apparently. So over here are those results. The top names PP, EC, TT, DR refer to different bacteria whose spent media was experimented with. Over here is the picture of what those spent media have, those proteins for example, and over here the carbonylation of proteins after exposure to radiation was examined using spent media of different organisms. As you can see in first 3-4 cases, there is a lot of carbonylation, but the DR is Deinococcus radians, you hardly see any carbonylation, indicating that that spent medium is protecting proteins substantially from radiation damage and carbonylation. Then they used the restriction endonuclease BAMH1, which is one of the most radiation sensitive restriction enzymes apparently, and they tested the ability of such spent medium to protect BAMH1. And as you can see here, 0 is in the absence of radiation, and it loses that activity from other organisms very soon. But in case of DR or Deinococcus radiodurans, you can see the restriction activity is present for a considerable length of time. Subsequently, they used even older media and found that even after 66 days at the end, if they used the spent medium from Deinococcus, it protected BAMH1's restriction endonuclease activity, suggesting to them that this spent medium is a very potent source of antioxidants. So they fractionated it to find what it has, and they found basically it has several amino acids, but 10 of them most abundant. They are listed at the bottom here. They also found it was rich in phosphate buffer, and they found it was very, very rich in manganese ions. Manganese is a very important requirement for growth of Deinococcus, including for its radioresistance, and all that it lets out. So they synthetically, artificially prepared these combinations and found the best combination was what is listed in the lower row as number 3. 25 millimolar of phosphate buffer, 3 millimolar of a decapeptide, and 1 millimolar manganese chloride. This is their magic decapeptide. So this whole thing, they call it MNDPPI, manganese, decapeptide, and phosphate, which is now regarded as the most potent antioxidant protein solution available from Deinococcus, and is being used by different people for different purposes. I'll just show you one example and close this talk, which has to do with ability to produce vaccines. In case of COVID also you saw, or many other places, it's desirable to produce vaccines to the whole organism and all its epitopes. But such organisms are invariably pathogenic, and therefore you may have wrong things coming into the vaccine, making the vaccines unsafe. So one thought of irradiating that organism with radiation to destroy it, to prevent its infectivity, pathogenicity, viability, but at the same time the epitopes which are required for producing antibodies should not be destroyed. And that's the result here shown. This is with bacteriophage lambda, which is not a pathogen except to E. coli. So what you see there at the top is the green curve is MNDPPI. It survives better and better in MNDPPI and dies very earlier. But then over here, using a DNA marker, you can see in the absence of MNDPPI and in the presence of DPPI, you can see by 20 or 25 kilogray, the virus is completely inactive. It has no viability anymore. But then you look at its epitopes using either Kumasi stain or specific monoclonal antibodies to the head and tail proteins. These are the two proteins you are seeing there, and in the presence or absence of MNDPPI. So what you can see there, conclusion, it says that the genome damage and killing can be uncoupled from epitope destruction in response to high doses of radiation if you do radiation in the presence of MNDPPI. So this was the initial result, and now what people have done, have tried to use very serious human pathogens, sometimes biological warfare agents, and even polio vaccines they have produced using MNDPPI, and that's the last slide here. This is the example of VEEV, which stands for Venezuelan equine encephalitis virus, a very deadly virus, lethal to not only humans, many other mammals also, has been a candidate for biological warfare. No antidotes are available, but it was thought if a vaccine can be produced against this, against the whole virus, it could be very useful. And what this slide shows you is that if it was irradiated in the presence of MNDPPI, the middle western blot using monoclonal antibodies, you could get complete destruction of the virus, but the epitopes survived up to about 30 or 40 kilograms. Now using this, an antibody, a vaccine has indeed been produced for this. And therefore, this is the future of this. In COVID times also, it was very actively debated whether it could be done. But first, a vaccine had to be available, which has now become available. It may be easier to produce a more safe vaccine, not only for COVID, for any other deadly virus, by using the whole organism and its epitopes for producing antibodies. So with this, I think I have given you some idea of some non-nuclear applications as well. I will close this presentation by showing the last slide, which is to acknowledge all the colleagues who have worked with me on Deinococcus. The green ones are those whose work I have presented to you today, including Dr. Hari Mishra, who has been inducted into INSA this year and will be getting his fellowship on the last day. I also thank all those who contributed a few strains and vectors to our work from abroad. And lastly, grateful to everyone whose work I have cited today. And last but not least, thank you all for your patient hearing. Thank you.